Myself, Dr. JIBRAN Ahmed presents to you Simply Pathology, and today we are back with a very important session. Today we are going to discuss about a very important exam topic, that is gene therapy. Now it is very commonly asked as a long answer question in your exams. And in the last 5 to 6 years, a lot of clinically approved gene therapies are now available. So there is a growing importance revolving around gene therapy. So please listen till the end of this lecture, because there is a very high chance that this will be asked in your theory examination. Okay, so along with gene therapy, in the subsequent lectures, we are also going to discuss some important topics like gene editing. Okay, that is the CRISPR technology, as well as the CAR T-cell therapy. Okay, all these three can be separately asked as a long answer question in your exam. Okay, so in today's topic of discussion, we are going to start with the gene therapy. So let us begin today's topic without wasting any time. So what is gene therapy? It is the medical technology which is aimed at producing therapeutic effect through manipulation of gene expression or alteration of biological properties of the living cell. So I will repeat. The most important definition of the gene therapy is that it is a medical technology aimed at producing a therapeutic effect through manipulation of gene expression or alteration of biological properties of a living cell. So over here, what we are doing, that we are bringing on changes at the level of the genome. So changes are brought at the level of the genome, okay, so as to provide a therapeutic effect. Okay, now gene therapy, if you see, it can be subclassified in multiple ways. So over here in today's lesson, we will see that there are three ways in which gene therapy can be classified. So the first and the most important way in which gene therapy is classified is on the basis of compensation for genetic alteration. It can be of two types. There can be a gene transfer therapy, and then there is a genome editing or gene editing. So this gene transfer therapy is the term used whenever there is an introduction of a new genetic material into the cell. Then we are using the term gene transfer therapy. And whenever we are modifying the existing DNA or the genetic material within a cell, we are using the term genome editing or gene editing. Okay. So first we will understand about gene transfer therapy. So gene transfer therapy is associated with introduction of a new genetic material. So it is associated with introduction of new genetic material into the cell, and it can occur under two settings. For example, an individual is having a mutated gene which is encoding for a defective or an absent protein. So by gene therapy, you can introduce a healthy copy of a gene which is encoding for a normal protein, and thus they can restore normal cellular function. So completely you have introduced a healthy copy of a gene. So normal proteins are now being encoded, and therefore normal cellular function has resumed. So this is one way. Sometimes even in the presence of a genetic alteration, okay, gene therapy can introduce an alternative gene which can encode for a protein which can compensate for the genetic deficiency and thus restore the normal cellular function. Okay, so whatever the clinical setting be, the gene transfer therapy is basically aiming at introduction of a new genetic material into the cell. The second important type of gene therapy, that is genome editing or gene editing, which itself can be asked as a long answer question in the exam, and this will be discussed in details in the subsequent lecture. Okay, so in the genome editing or in the gene editing, basically you are modifying the existing DNA within a cell. So no new genetic material is added. So over here, remember that no new genetic material is added. And remember, this technique, that is the technique of gene editing, allows for precise addition, removal or alteration of genetic material. So basically, very precisely at a very specific location of a genome, you can add a genetic material, you can remove a genetic material, or you can alter the genetic material. But over here, remember, no new genetic material from outside is introduced. Remember, unlike the gene transfer therapy, in the gene editing, no new genetic material is added. Remember this point. Okay, I hope you have understood the basic classification of gene therapy as a gene transfer therapy and genome editing or gene editing. Okay, now the next way in which you can classify gene therapy is on the basis of in which cells you have introduced the gene therapy. So there is a somatic cell gene therapy and then there is a germline gene therapy. As the term itself is self-explanatory, over here in case of somatic cell gene therapy, you are introducing the genetic material into somatic cells, okay? So the therapeutic genes, they are transferred to any cell of the body other than, except, okay, other than means except, leaving, okay, except the gametes, germ cells, gametocytes or undifferentiated stem cells. So over here the therapeutic genes are transferred to any somatic cell of the body except the gamete, except the germ cells, except the gametocyte, except the undifferentiated stem cells. So therapeutic genes are not introduced at these level, at the germ level. So therefore, by doing this, the changes are only made at the level of the patient or at the level of the individual, and such changes are not inheritable by the offspring. This is very important. So over here, the therapeutic DNA is used to treat the disease, and basically single gene disorders or monogenic disorders are good candidates for this somatic cell gene therapy. And examples in conditions of immunodeficiencies or thalassemias, hemophilia or cystic fibrosis, these are single gene disorders. So these are good candidates for somatic cell gene therapy. So I will repeat, in case of somatic cell gene therapy, remember the therapeutic genes are introduced in the somatic cells of the body, okay, other than the gametes, germ cells, gametocytes or undifferentiated stem cells. So they are introduced in any cell except these germ cells, okay. So they are limited to the patient level or the individual level, they are not inheritable by the offspring. Whereas the germline gene therapy, as the term is suggesting, over here the germ cells are modified by integrating functional or therapeutic genes into their genomes. So over here what will happen, that whatever changes are made at the level of the genomes of the germ cell, all cells in the organisms will contain the modified gene, including the germ cells, okay, including the germ cells. Therefore, the changes are inheritable. So they will be inheritable, okay, they will be passed on from one generation to another. So I hope this is crystal clear to everyone. So somatic cell gene therapy, wherein the therapeutic genes introduced in all cells except the germ cells, and basically the somatic cell gene therapy, they are not inheritable. Whereas the germline gene therapy, if you see, the therapeutic genes introduced basically into the germ cells, okay, and all cells in the organisms will contain the modified gene, and the changes are inheritable over here in case of germline gene therapy. And the third way of classifying the gene therapy is, now coming to the third way in which we can subclassify the gene therapy, is that gene therapy can be of two types: in-vivo gene therapy and ex-vivo gene therapy. Now what is that? Before we understand the concept of in-vivo and ex-vivo gene therapy, we first have to understand the concept of vectors, okay. So remember, now suppose over here there is a particular therapeutic DNA, okay, of interest. And basically this is the particular host cell and this is the nucleus of the host cell. This is the host cell, okay. And basically this is having a faulty gene. So we want to introduce this particular DNA over here. So how do you do so? You have to do it with the help of a vector, okay. So basically in gene therapy, vectors are important for delivering genetic material to the target cell. And these vectors can be of two types. They can either be viral vectors or they can be non-viral vectors. So viral vectors includes, most common ones which we use is the lentivirus, that is the retroviruses, adenovirus, the short form is ADS, and adeno associated viruses like AAVs, okay. So remember these short forms because later on we are going to use them, okay. So the viral vectors includes lentivirus, that is the retrovirus group, adenoviruses and adeno-associated viruses. Then the non-viral vectors include injection of naked DNA, oligonucleotides or inorganic nanoparticles with the help of electrosonoporation or with the help of a gene gun. So over here a non-viral vector is used for introduction of genetic material, okay, inside the host cell. Now one advantage of the non-viral vector is that these technology, this non-viral vector, okay, they can be scaled to to produce a mass production of vectors can be produced, and the process can be carried out at a very high level, okay. So it can be scaled, and also one advantage is that because they are non-viral, so they do not induce any kind of immunogenicity, so there is a reduced immunogenicity. But one bad thing is that the rate of transfection, that is the introduction of the DNA into the host cell, the rate of introduction is very less. Okay, so there is a reduced transfection with them and reduced gene expression. So as a result, the overall efficacy with using non-viral vectors is reduced or is less. So more on that we will come on later. So right now you just have to understand that for the introduction of a DNA into the host cell, we require a vector. Vector can either be viral or it can be non-viral. Viral vectors include lentivirus, adenovirus, adeno-associated virus, and non-viral vectors include naked DNA, oligonucleotide nanoparticles, which are introduced into the host cell by the process of electro or sonoporation or with the help of a gene gun. Okay. Now we are going to understand the subclassification, the third subclassification of gene therapy, that is in vivo and ex vivo, and I will also explain you with the help of diagram, so please stay tuned. So first of all, in vivo gene therapy means wherein a vector, typically a virus, is introduced into the patient, okay, that is in vivo, passing the genetic material into the patient's cells. That is what is the meaning of in vivo, wherein the vector, typically a virus carrying the modified or therapeutic DNA, is introduced inside the patient's body, okay, in vivo, passing the genetic material into the patient cell. Now if you see, in vivo process is far simpler, as harvesting of mitotic cells is not required over here. Okay, so this is the in vivo process, whereas if you look at the ex vivo process, what happens over here, that the patient's own cells or cells from any allogenic source, they are extracted and cultured outside the body. That is why the term is ex vivo. Okay, now to this cultured cell, whatever cell has been cultured, that is the patient cell that has been cultured, to this cell the therapeutic gene is inserted, okay, into the viral DNA. The therapeutic gene which is inserted into the viral DNA, that is forming the transgene, is added. I will show you with the help of diagram. Patient's sample target cells are genetically altered with the transgene, and once this alteration takes place outside the human body, they are reintroduced into the patient's body. Okay, now one advantage of this method is that this ex vivo gene therapy is better tolerated and less associated with severe immune response. So over here, in case of the in vivo therapy, what is happening, that is a vector, that is a virus which is containing the therapeutic gene. It is introduced inside the patient's body. Whereas in the ex vivo process, what I am doing, I am taking out the patient cells, okay, out of the body, then I am culturing that, okay, and then to that patient cell I am introducing the therapeutic gene, okay, which is combined with the viral DNA, okay, and then it is basically added outside the human body. So the patient's target cells are altered with the transgene. Transgene is nothing but the therapeutic DNA, okay. So basically the patient's cells are genetically altered with the transgene, and all this thing is being done outside the human body. Once alteration occurs, they are reintroduced into the patient's body. Now remember, the ex vivo gene therapy is basically better tolerated and they are less associated with severe immune response. Now I know that you have understood only 20-30% of this. So now I am going to make you understand with the help of a diagram. So if you see, in case of in vivo gene therapy, this is the in vivo gene therapy. So this is basically a viral vector. What is this? This is the viral vector, and over here we have taken the adenovirus. This is the adenovirus viral vector, okay. Now this adenovirus, if you see, now this is basically the viral vector. It is having an outer capsid. This thing is your capsid, okay, and something which is projecting, that is known as the penton, okay. And what you see over here, what is this thing? You can see this particular DNA. Now this DNA is basically a modified DNA. Okay, so this DNA, a part of the DNA you can see is blue color, and this blue color DNA, it is the modified therapeutic DNA which is incorporated into the viral DNA. This entirely is the viral DNA, and some part of the viral DNA, if you see, it contains this therapeutic or modified DNA, okay, and this entire thing, this entire modified DNA which is incorporated into the viral DNA, we call it as the transgene. We call it as the transgene. Okay, Now what is happening, that this is the host cell. Now this viral DNA is entering the cell membrane, and as it enters, the virus is packaged into a vesicle. The virus, it gets packaged inside a vesicle. They form a vesicle containing the virus. So the increased acidity inside the cell causes release of what is known as the penton. As I told you over here, these spiky projections, they form the pentons. So increased acidity inside the cell causes release of pentons, which leads to breach of the vesicle. The vesicle dissolves or breaches or because of the release of the pentons. And once the pentons are released, now the viral is free, and the viral comes in contact with the nucleus, wherein at this level the virus disassembles and delivers the modified DNA. As you can see over here, the virus can, as you can see, this virus over here has disassembled and it has released this particular transgene or the modified DNA, which is already incorporated into the viral DNA, that is forming the transgene. So this virus disassembles and releases the transgene inside the nucleus of the host. Okay, and what you can see over here, this is basically the transgene that we have introduced. Okay, the therapeutic gene. And you can see over here the faulty genes. Okay, the faulty genes of the host is there, or the defective gene of the host is there over here. So there is a virus disassembly and delivery of the modified DNA into the nucleus of the cell, as you can appreciate over here. So I will repeat once again. As you can see, this is the in vivo gene therapy process. Over here, the modified DNA, that is the transgene, with the help of the viral vector over here, is basically introduced inside the host cell. So this is the in vivo process. It is taking place inside the patient's body. So as the virus is introduced, it is packaged into vesicles, and because of the increased acidity inside the cell, basically the penton is released, which is basically clearing the vesicle or breaching the vesicle. Now the virus itself is going to the nucleus. It is disassembling and introducing this particular transgene or the therapeutic gene inside the host nucleus. Okay, as you can appreciate over here. Now, one important aspect that you have to understand over here is that the introduced DNA do not become integrated into the host chromosomal DNA. As you can see, this is the transgene. It is not combining with the host DNA over here. Remember this thing. The introduced DNA do not become integrated into the host chromosomal DNA. Rather, it remains separate, thus ensuring that the newly introduced gene functions independently without integrating into the host DNA. This is very, very important, and this is the in vivo process. And the in vivo process, as I told you, it is far more simple, as harvesting of mitotic cells is not required over here. So this is the in vivo process of gene therapy, wherein a vector, typically a virus containing the transgene or the therapeutic gene, is introduced inside the patient's body or inside the patient's cell. So this is the in vivo process. Okay, everything, the process is taking place inside the patient's body. Now, what is the ex-vivo process? As I told you, the ex-vivo process is more complex in nature. So with this help of with this diagram, I am now going to explain you what is ex-vivo gene therapy. So please pay a lot of attention over here. So what is happening over here, as you can see, that basically this is the therapeutic gene. Okay, and this therapeutic gene is basically inserted into the viral DNA, and this is the viral vector. So what happens over here, a transgene is created, that is, along with the viral DNA, the therapeutic DNA is basically attached. So this is forming what is known as the transgene. Okay, and therefore this is made ready. And over here, what happens, that whatever target cells are there, they are initially removed from the patient. So whatever cells of interest or whatever cells in the body which are not functioning properly, or whatever are the target cells of the host, they are removed. And once they are removed, they are cultured. Okay, so they are grown so that a sufficient quantity of cells are there. Okay, and then what is happening, that the viral transgene along with this particular target cell of the host, they are basically taken together. Okay, so the patient's sample target cells are genetically altered with the therapeutic gene in a very similar manner that happened over here, but the only difference is this is taking place outside the body. This is taking place outside the body. The patient's sample target cells are genetically altered with the therapeutic gene. So therapeutic gene, it now enters the patient's nucleus, the target cell's nucleus, and once they have gone inside the nucleus, these target cells are reintroduced into the body. So whatever changes or whatever things which are happening, they are happening basically outside the body. Okay, so remember this point. And then inside the body, the genetically altered cells, they produce desired proteins encoded by the therapeutic DNA. So ex-vivo gene therapy is performed with the genetic alteration of patient's target cells happening outside of the body in a culture. Target cells from the patients are infected with a recombinant virus containing the desired therapeutic gene. These modified cells are then reintroduced into the patient's body, where they produce the needed proteins that correspond to the inserted gene, and therefore they restore cell function. So once again, now I am going to just go through the ex-vivo process so that this becomes crystal clear for you. Over here, the patient's own cells, okay, are extracted and they are cultured outside the body. Now this is the target cell, okay, which is cultured outside the body. Now to this target cell, what we are doing, the therapeutic gene which is already inserted into the viral DNA, that is forming the transgene. So to this particular target cell, we add this transgene. Okay, and once this transgene is added, what is going to happen? A similar process that we have seen, that is the particular viral DNA is going to go inside the nucleus of the target cell, and therefore they will introduce that particular therapeutic gene inside the nucleus. Okay, after this genetic recombination occurs, the patient's target cells, which is now genetically altered, okay, is now reintroduced into the patient's body. Okay, so what is the advantage of this ex-vivo gene therapy process? It is better tolerated and it is less associated with severe immune response. Okay, so this is about the in-vivo gene therapy and the ex-vivo gene therapy. I hope this is crystal clear to everyone. Now, what are the approaches for gene therapy? So rather, I would just say that you should just know the basic definitions of these terms and what it means. So there are four approaches. One is the gene replacement, wherein the malfunctioning gene is replaced by fully functional gene. So malfunctioning gene is replaced by fully functional gene. Then there is gene silencing, wherein the toxic mutated gene is intentionally deactivated. And then there is something called as gene addition, wherein additional genes are introduced to enhance cell function. And then there is gene editing, which allows permanent manipulation of the patient's genome. So there are four approaches to gene therapy: gene replacement, gene silencing, gene addition, and gene editing. Now the first clinically successful trial, okay, that is the gene therapy trial, was led by French Anderson, Michel Blaise, Steven Rosenberg, using ex-vivo strategy to treat the patient suffering from adenosine deaminase deficiency along with SCID, severe combined immunodeficiency, that is ADA-SCID deficiencies was there. And it was the first successful gene therapy carried out in human beings. Okay, now we will discuss about the viral vector systems. Okay, and remember this viral vector system, it is an essential component, each with a distinct function. Now basically remember, this viral vector system is comprising of certain components, each having a distinct function. What are these components? Let us see. This viral vector, they have a protein capsid or envelope that we have already seen over here. Okay, in this diagram you can see this is the capsid or the protein envelope. Okay, so this particular protein capsid or envelope, it encapsulates the genetic payload, that is the transgene. What is the transgene? It is the viral DNA, which is also containing the modified or therapeutic DNA. Okay, this protein capsid or the envelope, it determines the cell or tissue tropism. What is the meaning of tropism? It means that this particular virus, okay, it has a likelihood to go and infect certain cells of the body. So on the basis of the nature of the capsid, it decides which cells or tissues of the body that particular virus is going to go and infect. That is determined by the protein capsid or the envelope. It also contributes to antigen recognition, which may trigger immune response. So whenever you are introducing any foreign object into the human body, it is going to trigger some kind of response, right? So it is this particular protein capsid which is responsible for inducing an immune response from the host. Okay, so that is the importance of the protein capsid or the envelope, which is one of the essential component of the viral vector system. The next important component is the transgene. It is the gene of interest which has to be introduced into the target cell. That is called as the transgene. It helps us to achieve the desired therapeutic effect in the host affected cells. And thirdly, we are having the regulatory cassette. Now this is the combined. Now for example, you have already introduced, right? You have already introduced a particular gene into the genome, but there should be some machinery which will regulate the expression of that transgene, right? That is known as the regulatory cassette, which is comprising combined enhancer or promoter region that controls stable or transient somatic expression of the transgene. That means when and how it should be expressed, that is basically controlled by the regulatory cassette, that is the combined promoter enhancer region, that is going to control how and when this particular transgene will be expressed inside the host cell. So this is the most important essential components, okay, of the viral vectors. Now viruses introduces transgenes into the host cells, tricking the host cellular machinery into transcribing and translating the transgenes, okay, which is basically carrying the gene of interest, okay. And most commonly these particular DNA or transgene which is introduced, it never integrates with the host DNA. Therefore these are non-integrative vectors, and more commonly we use non-integrative vectors only. Okay, they will not go and they will bind with the host cell DNA. Now let me just give you one example. What I'm trying to say in this line is that suppose this is a particular host cell and this is the nucleus of the host cell, and suppose over here this is the host cell faulty gene or faulty DNA, and over here we have basically introduced that particular transgene. What we have introduced over here is a transgene which is containing the gene of interest over here, okay. So what they are saying that this particular gene that is there, it will use the machinery of the host cell for transcribing. This particular DNA will give rise to an mRNA, and by the process of transcription, and from mRNA you will get the particular protein by the process of translation. Over here, by the help of transcription, they will get the mRNA, and by the help of translation, you will get a particular protein. So basically, for carrying out this process, you will require ribosomes, you will require certain enzymes. So who is going to provide? The host cell itself is going to provide all these things, okay? So this is basically meaning that how the viruses are introducing the transgenes into the host cells, tricking the host cellular machinery into transcribing and translating the transgenes, okay? And more commonly this is what happens. But in certain viral vectors, for example the retroviruses, it goes a step further by having their genetic material integrated into the host DNA. These kind of viral vectors, they are called as integrative vectors. Why? Because over here whatever the transgene is there, it is getting integrated into the host DNA, okay. So this is also one way. So again, vectors can be non-integrative or integrative vectors as well. Now over here, in short, we are going to discuss the three most important viral vectors. That is number one, adenovirus, that is ADs, which are basically used in 50% of the clinical trials. Then we have AAVs, that is adeno-associated viruses, which are present in 28% of the clinical trials. And then we are having the lentivirus or the retroviruses, which are basically utilized in 22% of the clinical trials. So first we will discuss about the adenoviruses or ADS. Now this adenovirus, basically this vector is derived from large and complex